Welcome, this is Mr. Edstrom, and right now I'm going to be talking to you about dihybrid crosses, and that would be involved in determining the probability of outcomes for crosses involving two traits as opposed to just the one trait, which monohybrid crosses determine. It all boils down to what Gregor Mendel was figuring out here, pictured with all his monastery buddies. He happens to be looking at a specimen of a pea plant here. These guys don't seem to understand his fixation. At any rate, there he is again, and this is just to kind of remind you that it all boils down to that dominance and recessiveness that we're going to be looking back at again when we look at these dihybrid crosses. When we're looking at these, we're again talking about dominance and recessiveness. And so if we have a replicated chromosome here, what is not indicated and what would be correct for me to do then is just to kind of indicate that same copy over on this. So this would be a dominant tall allele in this case with the examples that were given here. And over here there would be a recessive dwarf allele indicated there. These would be replicated, and those would be replicated of that one, and these would be homologous chromosomes. And this is just to kind of remind us about the dominance and recessiveness, that if you had a heterozygous situation like so, with a big A dominant tall allele and a recessive short allele, the combination of those two things will give you a tall plant. You'd need two recessive alleles in order to get the dwarf or the short plant. This is really where we can start looking at the application of the segregation and the independent assortment. We've talked a lot about it in just terms of regular old meiosis, but here we'll be able to see it in terms of the passing on and probability of two traits. So anytime we're talking about meiosis, we're always going to be talking about two sets of homologous chromosomes, the paternal and the maternal, having replicated themselves already, and they are going to segregate. Each is going to go into a separate cell, and that's going to end up looking like this and like that. And they could separate, they will separate independently of how these two might separate. So you might have both maternal and paternal chromosomes going into the same cell, or you might have had them arranged a little bit differently during meiosis 1 so that they sort themselves independently. So you have one maternal and one paternal go into that cell, and then likewise over here for this cell. And you'd end up then with a 25% chance of getting any one of these different combinations, and that will be what we use when we do dihybrid crosses. So any one of these has a 25% chance of happening with these combinations of chromosomes in the first place, if we just had a total of 4 chromosomes in a cell. When we start looking now at specific traits, we'll use the smooth characteristic and the color yellow or green characteristic. The smooth and yellow are dominant traits, and again these are found on 2 separate sets of chromosomes. And so when you have a smooth yellow seeded plant crossed with a wrinkled green seeded plant, you will end up with no other possibility other than getting one of these little s's combined with one of these little y's in the gametes, and you'll end up with something heterozygous for both. And we'll talk more about how that works. But what we want to look at right now is the possibility, what the possibilities are of this heterozygous individual for both traits. So this is a heterozygous situation where you've got a smooth and a yellow plant, but it can now produce 4 different types of gametes based on how they segregate and independently sort themselves. So you could combine the big S with the big Y the big S with the little y, or the little s with the big Y, or both recessive alleles could sort themselves into the same gamete. And so you'd end up with something like that. And so if you had that as the starting point, and basically what you would do to do one of these problems is to set yourself up a 4x4 grid, because again, there's going to be 4 possibilities if you were going to be crossing one of these individuals with another individual of the same genotype, heterozygous for both of these traits. You'd have a 4x4 grid. So here would be one of those individuals, here would be the other individual, and the probability of the different types of combinations that you might get with both of those traits, the smoothness and the yellowness, or the wrinkledness and the greenness in this case for the recessive allele combinations here. And what you would do then is just do what we would do in monohybrids, is you would bring all those big S's and big Y's down into each of these squares, each of these big S's and big Y's across into each of those squares, and you'd end up with something like that if you were to do that same thing for each of the rows and each of the columns. And so what you would end up with then, if you look closely at the genotype, the phenotypes that would follow from this would be both dominant, homozygous dominant genotypes, and so you'd end up with both dominant phenotypes. If you looked at each one of those squares that had a big S, a dominant allele for smooth P's, you would end up with all of these smooth P's. And it turns out that if you combine all those at least one big S with a one big Y, 9 of them would end up being dominant for each trait. If you then looked at some of the other combinations you might get, here would be a smooth P, but it would be homozygous recessive for green. Same thing here. There's the dominant allele that would cause that one to be smooth, the dominant allele that would cause that one to be smooth, but homozygous recessive in all of those green cases. So far we've got 9 of the one type that are all smooth and yellow, 3 that are smooth and green, 3 that are going to be wrinkled, homozygous recessive for that smooth / wrinkled trait, and then homozygous, or excuse me, at least heterozygous for the dominant yellow trait, and then you'll have one little wrinkled green P. So homozygous recessive for both traits. And we would end up then with a 9:3 to 3:1 phenotypic ratio. Gregor Mendel, he did a few of these crosses, and this would be some of the data that he got, which led him to believe that there also is something mathematical probable going on with these dihybrid crosses. This led him to that independent assortment conclusion. He did not see anything like so, as simple as them all being linked together. We will talk about linked traits in the next video, but if there are any questions, make sure you write them down, and we will be talking about this a lot more on Monday. Thanks very much, and enjoy the rest of your weekend.